Welcome to the BioEngage webinar. Today, we are delighted to introduce Dr. Shihab Uddin, Assistant Professor at Department of Bioengineering. He got his PhD and MSc in Biochemistry and Molecular Biology. Dr. Shihab Uddin got his PhD in Biomedical Engineering from Kyushu University in Japan. He joined MIT as postdoctoral associate in the Department of Chemical Engineering. He is expert in synthetic chemistry, biomaterials, drug development, drug formulations, pharmacokinetics, pharmacodynamics, nanodrug delivery, immunotherapy, diabetes, cancer biology, and vaccination. His research interest is synthesis of lipid derivatives from natural biosources, microalgae, to introduce in diabetes and cancer targeting nanodrug delivery systems. Welcome, Dr. Shihab.
Thank you. Can you see my slide and to me?
Yeah, it's clear.
Okay, thank you. Good morning, everyone. Thank you, Dr. Farid, for your nice presentation. I'm Dr. Shihab Uddin, Assistant Professor at Department of Bioengineering in Kwara University of Petroleum and Mining. Before starting, it's not playing.
Is there a problem?
Just a minute, it's not playing. Maybe. Okay, thank you. Now it's playing. Sorry to interrupt. I would like to introduce myself first because you can understand what is my background and what is my research interest. I have completed my BSc and MSc major in biochemistry and molecular biology from Jahangirnagar University, where I focus my research on the extraction and purification for anti-diabetic oral drug delivery system. Then I focused the animal study for the anti-diabetics property. Then I have joined in the largest pharmaceutical company in Bangladesh and worked there as a research scientist. Then I privileged to join at Kyushu University under the mixed scholarship to continue my PhD, where I focused my research on the synthesis chemistry, purification and extraction. Then I used this extraction lipid-based biomaterials in nanodrug formulations targeting to the cancer. Then I joined at MIT as a postdoc fellow. My research was the nanodrug formulation for human antibody, lipid nanoparticle, and mRNA delivery for nanovaccine. Now I joined in KFUPM at the Department of Bioengineering. Okay, let's start to talk about today's science. Today's topic is the lipid-based ionic liquid and novel biomaterials in cancer-targeted nanodrug delivery system. What is the ionic liquid? Ionic liquid is a solvent that consists entire of ion. Its melting temperature is below 100 degrees centigrade. That differentiated the ionic liquid from the molecular solvent and salt solutions. In 1914, first Paul Warden described about the ionic liquid. So the research of ionic liquid is not too old, it's quite new in the field of science. In the ionic liquids, cation and anion can combine a multiple different ways to optimize the property of ionic liquids. Here the property of ionic liquids and use of ionic liquids in multiple fields. But our target to use of ionic liquid in pharmaceutical industry, especially in the drug delivery system. How this ionic liquid incorporated in the drug delivery system? In pharmaceutical formulations, different types of material used as an excipient. Those ionic liquid can act as a good excipient for drug formulations. Besides that, this ionic liquid can involve in the drug delivery system, such like as to make API-ILs or dual or multiple functional API-ILs, which increase the stability of the drug molecules and also increase the solubility of drug molecules. Why? Ionic liquid mediated drug delivery system is needed for the human being. For solubility issues, if you consider that in the market, up to 40% drug is available. In the sense, more 60% drug are underdeveloped. or in the research stage due to the lack of solubility, stability, and bioactivity. Ionic liquid can play a key role to increase the solubility of different drug molecules that can increase the stability in the formulations as well as increase the bioavailability or biocompatibility of drug molecules so that ionic liquid can be a significant biomaterials that can play important roles in the drug delivery system. Aiming this, we develop a lipid-based biocompatible ionic liquid. Why we choose lipid-based ionic liquid? Because our target to use this ionic liquid in transdermal drug delivery system so that we use a cationic part that is for a lipid phospholipid derivatives called EDMPC that is a biocompatible, and we use anionic part and amino acid. This is also a source of biomaterials. Normally, if we want to develop any biomaterials, definitely we should need both two parts of chemical source should be from biomaterials. So that we choose cationic and anionic part both from the biocompatible sources. Besides that, our target is transdermal drug delivery system. Our skin favors the hydrophobic mediated drug delivery system. So we choose lipid for the drug delivery system. Those are the lipophilic in nature. They have different types of routes of drug administration in our body, just like as oral, inhaler, transdermal. Among them, the transdermal drug delivery system is the most prominent because in our body, 50% part of our body is skin. So it's a very large and very well populated part for drug delivery system. Normally, in drug delivery system, they have intercellular, intermuscular routes. We use this ionic liquid in drug delivery for the intercellular or intermuscular drug delivery systems route. For this reason, first we developed lipid-based ionic liquid. How we synthesize this ionic liquid? First of all, we take the MPC lipid powder that's react with ethyl difluoromethyl sulfonate to prepare the EDMPC chloride. It is the cationic part of ionic liquid. Further, this cationic part of ionic liquid reacts with the different types of amino acid, such like as linoleic acid, oleic acid, and stearic acid, and thus we prepare the lipid-based ionic liquid. Okay, we prepare the lipid-based ionic liquid. How we confirm that this lipid-based ionic liquid prepared properly? So that we need to characterize these new materials. First of all, we characterized with TLC and found that the synthesized ionic liquid show an identical peak on the TLC plate. Further, when we confirm that they have some new materials developed, then we can again characterize this biomaterial using the NMR and also investigate that our synthesized ionic liquid that's considered the cationic part is acid, they didn't contain any acidic protonic peak in NMR, which is represented that our new synthesized ionic liquid doesn't contain any the carboxylic part. So this is one our targeted compound. Then further we characterize by FTIR and mass spectroscopy. Both cases we found that our targeted formulation shows identical peak what we are wanting. Then further we investigate the therapeutic index of this new ionic liquid and found that different ionic liquid, EDMPC, linoleic, that means linoleic acid based ionic liquid, oleic acid based ionic liquid, stearic acid based ionic liquid, their refractive index are near to similar because of their physiochemical properties near to the similar but quite different. Then we investigate the thermal phase behavior of our new synthesized ionic liquid. And we investigate that they have different melting point and different crystal point and phase transitional point. Due to the structure of anionic part, oleic acid, linoleic acid, and stearic acid, they are different. So that based on their cationic anionic part, their melting point temperature and crystalline temperature are different from each others. Further, we confirm this phase behavior at different temperature, especially for melting and crystalline temperature. We use thermal phase behavior using the optical polarized microscope, where we maintain the temperature and found that EDMPC linoleic acid based formulation at 20 degree centigrade it's remain crystal, but when increase the temperature up to 30, then it's become liquid. That means in its melting point between the 20 to 30, such like as EDMPC chloride, EDMPC oleate, EDMPC stearate, they have different crystalline phase at low temperature, but after certain temperature increasing, their phase are changing from crystalline to liquid, which is remain the melting temperature. Then we correlated with each others, such like as optical polarized microscope analysis and physical appearance. Then we also investigate computerized static behavior between two molecules, such like as defined to change oleic acid, linoleic acid, stearic acid, that combined with the EDMPC and found that the EDMPC chloride, which is cationic part, their internal bonding are so far, as a result, their melting temperature is low. As a result, same linoleic acid and its oleic acid, their melting temperature completely low, so that they can easily melt it from the crystalline form to the melting point. But when we draw the 3D structure using the computational approach and found that stearic acid binding capacity of stearic acid with the cationic, they are bind tightly. As a result, their melting temperature also increase. Those theory results supported each others and confirm that our synthesized product is as we are wanting. Now, further, we confirm the purity of this newly synthesized biomaterials or ionic liquid using the chemical analysis, where we show the how much amount of carbon, hydrogen, nitrogen, and moistures content. And here we found that our synthesized compound contains a little amount of water. It is called intermediate water. For the biomaterial phases, sometimes intermediate water increase the bioavailability and also increase the solubility property of this newly synthesized ionic liquid. Then we investigate the solubility of this ionic liquid in different solutions, such like as polar, non-polar, oilic, and organic, inorganic, and found that significantly it is very interesting findings that our new synthesized ionic liquid of biomaterials are soluble in both polar and non-polar solvent, because the cationic part or synthesized biomaterial contain lipids which contain the both polar and non-polar. But this property of lipid can help to solubilize both organic or inorganic or polar or non-polar solvent. Then we are taking that, we are considered that our synthesized ionic liquid is biocompatible, but we need to prove it. For this reason, we use this ionic liquid in the human artificial 3D lab-side EP model cell line, using the MTT assay method, where we apply this ionic liquid after 24 hours and found that our synthesized ionic liquid is comparatively the safe, like IPM, which is the pharmaceutical accepted biocompatible solution, and our newly synthesized ionic liquid are safe as IPM or PBS saline. So we can say this synthesized ionic liquid is biocompatible. Now, first April, after synthesize this ionic liquid, we would like to check how it's active in drug delivery system. Aiming to this, we take firstly the peptide drug, while we use our synthesized ionic liquid in formulation to develop the nano drug delivery system. And we use the leuprolide. It's a model peptide drug to develop a nano formulations. Aiming this, first of all, we use our ionic liquid in cyclohexane, make an emulsion process in the oil medium. And finally, we prepared iodine oil nano dispersion solution. This solution is also known as a nano formulation or nano drug formulation, which contain a surfactant and post-surfactant. During our study, we found that our ionic liquid can wrap the drug particle to make a nanoparticle or use in nanotechnology. But how ratio is suitable for this formulation? We use different type of ratio, uh, half ratio, 1:3 ratio, 3:1 ratio, 4:4, and like these ratios, and found that only the equal ratio of surfactant and post-surfactant make a stable formulation. Based on this sorting strategy, we choose the best one formulation for our further study, where we show that equal ratio of surfactant and post-surfactant based formulation is stable, and it will be easily go forward with the drug delivery system. And further, we investigate that our ionic liquid can make formulation itself, but stability of this formulation increase when we use different type of post-surfactant. Aiming this, we use different type of post-surfactant to show which post-surfactant is better. And finally, we investigate that our ionic liquid-based formulation play a good combination with SPAN 20, and we go forward with the SPAN 20 and ionic liquid-based formulation in drug delivery system. Okay, when we prepare the nano formulation, we need to prove that it's a nano formulation or here some nanoparticles are forming. Aiming this, we characterized our new formulation using the DLS, dynamic light scattering spectroscopy, where we measured the particle size in our formulation. And found that our all ionic liquid-based formulation remain the same. Only ionic liquid-based formulation make nanoparticle, but particle size is not in nano range, it's micro range. But when we use the co-surfactant, it consists the nanoparticle and increase the formulation stability so that they are significantly decrease the particle size. And we know that when the particle size is decreased, then it increases the drug delivery rate through the skin. Further, first of all, we prove that they make nanoparticle. But what is their shape? So that we observe this particular shape using the TEM analysis. During the preparation of TEM, one step is the washing step, but our ionic liquid is fortunately soluble in both polar and non-polar solvent, so that during the washing process, the nanoparticle are destroy their own shape. So further, we investigate using the CLSM microscope and found that they have a lot of nanoparticles formed inside the IPM or inside the solution. And those nanoparticles are the size of those nanoparticles are similar to the DLS or TEM analysis. So we confirm that the formulation making the nano dispersion in the oil medium. Then we need to check how much drug are encapsulated in this formulation. Aiming this, we disperse this solution in methanol and acetonitrile containing solution and make concentration using the HPLC analysis. Ultimately, we measure here the amount of how much drug encapsulated this microemulsion or nanoparticle and found that near to the 100% or more than 90%, that means 97, 94, 91, more than 90% drug are encapsulated in the nanoparticle. So it's a really good approach for the drug delivery system to apply on the animal or body. Then another important thing is that to develop a formulation, how much they stable. And we found that we investigate this formulation in different temperature, low temperature, room temperature, and high temperature at up to different times. At low temperature, we found that our formulation making the aggregation, because lipid has a property at low temperature, they make aggregation. And thus they increase the stability at room temperature. Then we found that up to 19 days, this formulation remain unchanged. Then we applied this formulation in vitro, means out of animal, and found that our formulation significantly increased the drug delivery rate compared to the all others market formulation or in-house formulation. Then we also investigate the topical and transdermal delivery. Topical means how much amount of drug inside the skin or how much amount of drug inside the layer of skin. And we found that both topical and transdermal drug delivery system, our formulation increase the drug delivery rate or amount of drug delivery rate. Then we investigate the pharmacokinetics parameter of this formulation. Especially when we investigate the pharmacokinetics parameter, we found that the cumulative amount of drug in the body, in the skin, is the significantly increase in our F1, means our new developed formulations. Such like as what happened normally when use those drug in the skin, they increase the facility of skin delivery through this nano formulation, and found that our drug formulation that increase the shift of the skin structure which facilitated drug into the skin. It is the more than the highest amount of shifted. It's known that the drug which shifted the arrangement of skin which facilitated the drug delivery system. Our formulation increased the facilitation of this drug delivery system. Then we investigate the pharmacokinetics effect of this drug on the mice and found that our drug formulation gradually increased the effect of drug in the blood plasma for a long time. But marketed formulation, they increase the concentration of a drug certain time. After certain time, they decrease the concentration of drug in the plasma, which is indicate that our formulation is really good for the control drug release delivery system. Then we also check the bioavailability of this formulation and found that this formulation is bioavailable and non-toxic for the skin or other part of the cellular membrane. Here show that the skin image of when we apply our formulation, we found that no adverse effect in the formulation, which is the really good symptom for the bioavailability of this formulation. Then we use this formulation for the large size of drug delivery system. First of all, we use peptide, small size. Then we use large size of protein. It is the OBA. It is very large and model protein, and investigate the formulation stability as size like as previously. Then we use this formulation in the drug delivery system to investigate the skin irritation or the cellular toxicity, and found that our formulation doesn't impact any toxic effect on the skin, which indicated the formulation bioavailability, biocompatibility. Then we investigate the routes of drug delivery system, how much or how this drug delivery into the skin. Through the system, we found that our formulation-based drug are significantly increase the flow of drug inside the body compared to the market solution and the commercial surfactant-based formulation. Then we investigate the in vivo effect. We use different times of time-based formulation on mice and investigate that inside the mice, the rate of drug delivery system significantly increased compared to our market formulation. Then we further confirmed that's what is the route of this drug, and found that the routes of drug delivery system through the skin is the intracellular routes. Then we investigate the tumor immunoresponse. What is the immunoresponse of tumor in the mice? As a result, we developed a tumor in the mice. Then we applied our formulation and investigate that the formulation where we applied our new formulation that decreased the tumor volume rate as well as decrease the death rate of the mice, and also the body weight was the remain unchanged as the marketed formulation, which is represented that our formulation has a good impact to decrease the tumor volume, growth, and rate. Then we also investigate the immunoresponse of this drug. Aiming this immunoresponse investigation, we input our solution, input our new formulation in the mice. Up to the 21 days, we investigate the amount of different immunoresponse in the body blood plasma and investigate that our formulation and injected formulation are remain significantly increased, but quite lower than the injected formulation in sense of immunoresponse. Then we investigate what is the immunoresponsible cell are stimulated in the tumor. As a result, we cut the tumor and to investigate the immunoresponse of this cell inside the tumor and found that our formulation show the maximum immunoresponse inside the tumor microenvironment, which is responsible for the anti-cancer property. So that it's easily understand that our new formulation has a good impact to reduce the tumor volume as well as reduce the cancer properties. Then we also use the real drugs such like as insulin in diabetics. And we found that when we use insulin and found that the plasma level insulin or the drug related to our formulation is higher than the water-based formulation, because our formulation facilitated the control release drug delivery system. And we also check the bioavailability of this drug and toxic effect of this drug in the cell line as well as the animal model. In all cases, we found that this formulation is really good for this. When we check the immunoresponsibility of the drug using our ionic liquid, we found that the immunoresponsibility is quite low compared to the injection formulation. To aiming that, we choose the best surfactant and best oil media to make a boosted formulation using the adjuvant. Adjuvant is a FDA approved immunostimulator that can increase the immunostimulation in our body. They have 3 immunostimulator that's already FDA approved. One of them, the Impumod. Aiming to this, we developed a nano vaccine using the immunoresponse adjuvant and our targeted ionic liquid and the drug, that is the protein drug. And when we develop this for nano vaccine, then we investigate the transdermal delivery of this nano vaccine and found that the nano vaccine with different surfactant has different roles, but we choose the best one for this salting strategy and found best result applied in the anti-cancer mice to develop the vaccine. When we want to develop the vaccine, we need to know the prophylactic effect and therapeutic effect as it is. Prophylactic effect means we take the medicine in our body will develop the immune system in our body automatically after the inoculation of the vaccine in our body, if we don't affected by the disease. So that first of all, we inoculation our new formulation in mice body and investigated that they have increased the anti-cancer immunoresponsibility antigen in the blood plasma. Here is the when we use the immunomodule or adjuvant that increase the immunoresponse even the injection formulation. That means when we use adjuvant with ionic liquid-based formulation, they help to control release drug delivery system. As a result, the drug are long time stable in the blood and increase the immunoresponsibility in the blood. Further, we investigate the therapeutic effect. Okay, first of all, we investigate that they have prophylactic effect. That means we are not affected by disease, but in our body, we developed the immune system. It is the main theme of vaccine. Then we confirm that is it really working or not. First of all, we make disease in mice, then use our formulation on the mice, and found that same time we found that when use the formulation in the mice after affected by disease, the immunoresponsibility is significantly higher than the injection. It's really surprising output. Then we further investigate the molecular level of deep immunoresponse. They have some others immunomarker that is the really responsible for the reducing the cancer or reducing the tumor, and found that the control our formulation really shows the good result and increase the amount of the immune response in the tumor cell. And ultimately we found that they have good impact to increase the anti-cancer immune response. Then further we investigate that is it really active in the tumor microenvironment? Then we cut the tumor and investigate that yes, in the tumor microenvironment, our formulation also increased the response of anti-cancer immunity, which is indicate that our new formulation is named as the nano vaccine, which contain the lipid-based ionic liquid, drug, and adjuvant is really good and better than the injection. Now, okay, it's better. Is it really safe for the body? Because when a drug are absorbed in the body, they have different organelles which is associated with the drug metabolism, such like as liver, kidney, heart, spleen, lungs. Is there any adverse effect of this formulation on those drug metabolite associated organelles? Then we check this and found that surprisingly, our formulation doesn't make any adverse effect on the drug associated organelles. Based on this study, we can summarize that our newly developed LBILs is a novel biomaterial because it is very new in the field of ionic liquid research. And they don't have any adverse effect on the cell line as well as the animal model, which represented that this newly synthesized material is a biomaterials. Then we investigate that this is the first developed ionic liquid which are soluble in both polar and non-polar solvent. It's really interesting. Normally, ionic liquid are soluble in polar solvent due to their ionic properties. But interestingly, our newly developed ionic liquid are soluble both in polar and non-polar solvent. And this ionic liquid boosted the formulation of nanoparticle that help in the nano drug delivery system, aiming to the cancer and diabetes, whatever we need. And this LBIL associated nano drug formulation significantly boosted the immune response in the animal model and also helped to improve the anti-cancer nano vaccination. So more studies such like as clinical trial or nucleic acid delivery. My previous lab also working on this. And hopefully this will be a great achievement if it's come to the market or if it's become helpful for the human beings. Thank you, everyone. Thank you for your time.
Thank you, Dr. Shihab, for this nice presentation, very valuable information. So does anyone have a question? Please write it down or just raise your hand so I can unmute you to ask your question verbally. So if we don't have questions, if you allow me, I would like to ask some questions because actually I have worked in something similar to the principle of this work and drug delivery.
Yeah.
I'm wondering if you if this carrier or a nano carrier, it's work for a specific type of cancer, or it can be used for like many different types of cancers, because the microenvironment for one cancer is different than the other.
Okay. Thank you for your question. Ultimately, the different types of cancer or anti-cancer property depend on which drug you are selecting. Such like as for every cancer, not a small one drug is effective. If you use such like as breast cancer, they have some drug that's used in the breast cancer treatment. But when you use that breast cancer targeted drug, then apply on the applied through the skin, then it will be effective against the breast cancer. If you use the colon cancer like this, then you choose the drug. It's depend on the effect of drug, depend on the which drug you are choosing or which type of cancer you are developing. In animal model, if you develop any specific cancer, you should provide the cancer specific drug. Here, and people use as a carrier. Let's increase the delivery rate, even increase the delivery then the injection formulation.
How we can ensure this composite, the carrier with the drug will reach to the destination and to avoid premature release
Yes.
system.
If you use orally, then we have fast pass metabolism by which maximum drug can destroy in the GI tract metabolism. But if you use this drug through the skin, to avoid those disadvantages of oral drug, we use transdermal drug delivery system. That means we use drug through the skin, which has no the fast pass metabolism effect, so that GIT will not affect or affect your drug. Directly you use your drug on the skin and drug absorbed through the skin inside the muscle or blood cell.
Okay.
And they have different types of immune response. I already show that IgG, IgA, IG2A, CD7, CD8, CD9. They have different types of markers inside the body, inside the blood especially. Those are responsible for the anti-cancer property. If you use your drug and in your body develop those type of antigen or antibody, that means your body has such capability to fight with the cancer.
Yeah. Okay. And for designing the carrier, when we go for a smaller size, like nanoscale, we increase the surface area so we have a possibility to load a much amount of the drug.
Yeah,
is there any side effect or negative effect when we go for a smaller size?
Yes. For transdermal delivery or any drug for the liquid formulation, especially when it's make a small nanoparticle, it will be better for absorption. If you use through the skin below the 500 nanometer, only permitted through the pore of skin. If you make the particle size more 500 nanometer, that's not really good for the transdermal delivery. So small delivery with the large number of or maximum amount of drug is the best formulation.
Okay, I got it. I got it. So from our audience, do you have any question? So this is the end of today's webinar. Thank you, Dr. Shihab Uddin, for sharing these valuable insights, and for our audience for your attendance. And we look forward to seeing you at the upcoming webinar. Thank you.
Thank you.